This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 405, recorded on September 2nd, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. This episode is also sponsored by Drobo, a family of safe, expandable, yet simple-to-use storage arrays. Drobos are designed to protect your important data forever. Visit drobo.com to learn more. And just to remind everyone, we're giving away copies of the recently published book, Virus, an Illustrated Guide to 101 Incredible Microbes by Marilyn Rusink. Keep on listening to find out how to enter. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
How have you been?
Been well, thanks.
Not that I don't know, right?
No, you come in every day. We're working together on a website, which we're about to announce soon. So we've got something exciting waiting in the wings.
It's 27 Celsius and sunny.
Yep.
The clouds are moving in. This morning there were no clouds.
It's actually a really nice day out today. The humidity is very tolerable.
46%.
And the weather is, I wish it stayed like this all year, but it won't.
We have some active weather alerts. We have some high
high
winds and flooding coming in.
Got some stuff coming up the coast.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Hey, Kathy.
Welcome back.
Thanks. It's gorgeous here. It's 72 degrees,
Look at
sunny. That's 22 Celsius. Just absolutely perfect.
I miss the hot and humid weather, Dickson. I really do. I love that. Also joining us from western Massachusetts, Alan Dove.
Good to be here,
Yeah.
and it is absolutely gorgeous here.
Yep.
It's 27°C, yeah,
Look at
a few
that.
puffy clouds in the sky. Lovely, lovely weather.
Yeah.
I'd say it was gorgeous here, except Dickson's sitting across from me.
Oh, be nice to Dick.
Come on, Caleb's listening.
Hey, Dickson, there's an interesting cloud. There are high clouds,
Cirrus.
which are completely covering the blue sky, and then there are very low, puffy ones.
Correct.
You don't often see the combination, do you?
It's fairly common.
Anyway, there's a hurricane moving up the coast. It hit
There is.
Florida last night, and Rich Condit wanted me to let you know that his weather is hurricane. He had like winds up to 50 miles per hour,
All right.
but he's okay. They just lost power.
Yeah.
And this is supposed to move up the coast. And yesterday we did a TWiM with Michael Schmidt, and he's in Charleston, right on the coast of South Carolina. He said
zapped.
they're going to get hit.
Yep.
And then it's supposed to come up here to New Jersey, and we're going to have winds up to 55 miles an hour on Monday with flooding, coastal flooding.
But no rain, I understand. So I don't know. We could use the rain.
I have to secure my boat.
Right.
And so does Griffin, Dickson.
He does. That's true.
So anyway, so that's the weather.
Yeah.
And Kathy, did you have a good travel in Europe?
Oh, it's great.
Yeah?
Went to the International Adenovirus meeting in Barsinghausen, which is a place of a famous soccer camp where many famous teams went and famous players all the way back to Pele. And the meeting itself was good. It was a really nice venue. We had some good social events and good science. And then I traveled some in Berlin and Dresden and then in Yorkshire.
Nice.
Cool.
Do you have a pudding in Yorkshire?
Yeah, well, if you take pudding to mean dessert, yes.
Good.
All right, we have a few follow-ups. We have an email from Jason Ladner, who was the first author on the paper on the multi-component animal virus, that genome that we did last week. Hi, Vincent. Comes from, he has a military email address, so it's surrounded by classification unclassified. Thanks so much for your fantastic coverage of our manuscript, a multi-component animal virus isolated from mosquitoes, in this week's episode of TWiV. I'm writing to provide an explanation to the question of why we decided to do the dose response curve in the first place. The dose response analysis was actually conducted expressly to test for a multi-component organization. We played around with some other ideas using modern technologies, but in the end we felt that this tried and true method was most appropriate. Of course, this begs the question of why we thought it was worth testing for the nature of segment packaging in the first place, which is what the question we asked, right? We said, hey,
that's
first
what it
I'd say this is in large part due to my own naivete, having only recently started to study viruses. When I first assembled this genome, I had very few preconceived notions about how the segments should be packaged. I was also tipped off early on by the lack of segment 5 in 2 of 6 isolates. Although a multi-component organization is not required for segment loss, it certainly provides a straightforward mechanism for the establishment of an infection with a subset of genome segments. And then, of course, once the electron microscope data came in, the small particle size was another good clue that the virus could be multi-component. And to their credit, my PI, Gustavo Palacios, and my colleagues at USAMRID were very receptive to this idea and helped enormously to properly test the hypothesis. Jason. P.S. I'm happy to report that we were able to get Figure 3 considerably reduced in size for the final version of the manuscript. The figure definitely didn't need a full page. Classification unclassified. Caveats none.
What I didn't hear you mention on the episode was Figure 4. And so because he talked about Figure 3, I had to go back and see what was Figure 3. And Figure 4 is gorgeous. It's the one where they show multiple combinations. One's green, red,
we did.
blue.
We mentioned
and
it. Fish,
did you? Okay.
Fish.
Yeah. It's absolutely beautiful.
But if you could read a stoplight, you could see this paper.
Well, Dickson, right. I can't believe it. You're actually on top of things. Oh my gosh!
Why? I've always been there.
No, I just haven't observed this thing that you don't pay attention.
Yeah, I fake you out sometimes like that, don't I?
We have an email.
Good job, Dickson.
We have an email from Tarwin, who's a longtime listener and occasional writer. Hi, TWiXers, writing from quote sunny unquote San Francisco, the only town in the U.S. that has been in the mid-60s for the past few weeks. This actually just came in this week. He's writing about advertising. I know you have had some backlash against advertising. I'd like to weigh in. It's always tricky moving from a model that is totally free to one that is either paid or ad-supported. One thing you may look into is making sure that your advertorial content is separated from the main show itself. Many podcasts do this by adding consistent background music behind these sections. Another thing that may annoy people is that you are championing your advertisers, Drobo, for instance. This may seem to some people like paid-for bias. I realize you actually do use and enjoy their products, but it can get confusing, especially when others in the group weigh in, and it is tricky to tell that you're actually listening to advertising. The podcast Startup did a whole show on how they have to walk that fine line, and it's worth listening. Personally, my only problem with advertising is in shows that are single interviews. The interview with D.A. Henderson, for example, is of historical record in some ways. And it's a pity to break it up like that. I'd suggest adding a version to your stream or just on your site without the ads for the future. All right, let me address some of these things. These are important. Now, one of the things that makes podcast advertising so powerful is that the podcasters typically have a personal relationship with listeners. You know, you listen to that person every week or every other week, and you come to trust what they are telling you, and advertisers like that. So if we pitch a product, we probably use it or would use it, not like other forms of advertising. And I've heard this on many, many other podcasts. And in fact, if you listen to the podcast on the TWiT network, This Week in Tech, they always go on and on about how they use something and they love it and they make millions of dollars in advertising. In fact, Drobo wants us to talk about the product and our experience with it and so forth. And of course, Curiosity Stream does as well. So they want you to get personal because that's what differentiates podcast advertising from radio and TV and so forth. Now, I don't know many podcasts that add music in the background. I know one that it's called ATP, Accidental Tech Podcast, where they put a little jingle in. No, it's not right. Jingle in front of the ad, and then after the ad, sort of to distinguish it. We could do that if you thought that was an issue. But I don't want to take the personal out of the
paid personal.
What'd you say?
The following is a paid.
Yeah, right.
The paid promotion.
That's right.
Now, as for the interview, the D.A. Henderson. Okay, I wasn't sure if I had to put ads in that, and I emailed our ad agency. I haven't heard back yet. It could be that there are special TWiVs without a number, and they don't fall into the. If they don't, I won't add them. And in fact, I'll go back and edit them out of that and repost the file, because I agree with you. It kind of interrupts it. So those are my thoughts. I think that these advertisers come to us because we have this kind of relationship. They want us to sort of incorporate it into the program. I wouldn't advertise something that I didn't think was good. Now, CuriosityStream, before we took them on, they gave me a subscription and said, check it out. And I did, and I thought, I think this is pretty good, and there's nonfiction content, and people would like it. And the same with Drobo. Years ago, they sent us a Drobo. We checked them out, and we like it. So they've pitched other products to us, and I said, no, I don't want to do that.
Right.
I'm not interested. And so that's the layer of how editing, let's say, that a podcast can do as opposed to a radio, which has to take basically every ad. I wouldn't take, for example, ads from pharmaceutical companies, especially ones that make vaccines or antiviral drugs, because we're always supporting and defending them, and it would seem like we're taking money, just like the fellow in Florida, who took money from Monsanto, and he's pitching transgenic plants. He got really hung by that. So, I mean, so what do you fellows and ladies think on this show? Do you think we should put music in the background? Do you think we should put a little jingle at the beginning and end? What are your thoughts on that? I'm sorry.
Who wants to go first?
Or no one cares?
No, my own feeling about the ads are that they should come at a prescribed time in the show, probably at the very beginning, and then not be mentioned again.
Well, you can't put them all at the beginning, Dickson.
You only have two.
Yeah, but you have to intersperse them.
Yeah, but you do that together, though. You do mention both.
No, but they're mentioned. The thing at the beginning is just like a billboard. That's called a billboard. You say at the beginning of the show, these are our sponsors. Okay. And you'll hear more as the show progresses. And then the ones in the middle are called interstitials.
Yeah, no, I know what they're called. But
and then you have to do a little more talking, 2 or 3 minutes.
Yeah.
And I try and put them at places where there's a natural pause, right? Even in interviews, I try and do that when there's a pause, but that's harder.
No, I remember when we were interviewing Harold Varmus, you went to a commercial break, and you could watch his face cringe as you were talking.
No, he wasn't cringing when I advertised PLOS pathogens.
No, him. No, of course he loved that part. But you didn't get paid for that, though, did you?
Of course I did.
PLOS pathogens paid you for that?
Yeah, what do you think paid for your dinner? And pulling everyone in, having everyone travel in. Yeah, they gave us money to support that episode.
I see.
Anyway,
right.
Kathy, what do you think? Any thoughts?
I don't listen to that many other podcasts, and the other ones I listen to, I don't remember them having ads.
Okay.
So I don't have much to compare it to, but it seems like, if you wanted to put a little jingle in at the beginning and end of each ad, I mean, it's more work, but I don't know. Wouldn't hurt, but I don't think you have to have music underlaying it. That'd be kind of weird.
Alan, I don't think
you need a designator like a jingle or anything like that, because it's obvious when you're going into an ad. Right. I mean, the words and the context of it, it's very clear that these are advertisements. They're not part of the program. Certainly, we're not taking ads from anybody that might pose a conflict of interest. That's just, journalism 101. So yeah, pharma companies, we simultaneously criticize and defend them depending on the topic. So that's not going to happen. There are a number of other podcasts that do this, obviously. My own personal model for podcasting has actually been an old school radio show that I listened to years ago when I lived in New York City. You can Google a guy named Vin Scelsa.
Yeah, I remember him.
Who did a late night. I listened to this all through graduate school. It was awesome. Every Sunday night I had my routine. I would listen to Vin, and I don't mean Vincent.
Yeah, I know.
I never listened to him. No.
Maybe you should have.
Maybe I should have. I would have gotten out in fewer than 6 years. Yeah. Or was it? It was more like 7. Yeah. Anyway, no, Scelsa. he did this very free-form show that ran for a couple hours usually, sometimes a little long, and he would have musical guests on. It was a music show. But he did ads, and he would do ads that were built into the show, and he would talk through the stuff, and it was always stuff that he was willing to endorse, but I'm sure it was very valuable to have the host of the show talking through it like that, just like we do.
Yeah.
So this is absolutely not a new thing. It was done back before podcasts or pods or any of that existed. And it doesn't raise any new problems. I see the concern that listeners have. They were listening to this podcast for a couple hundred episodes, and there were never ads on it, and now there are.
There were Drobo ads way back.
There were, yes, a couple of years ago there were Drobo ads, but if we want to advance this further and have things that we travel to and get the whole crew together for, then this costs money, and none of us are independently wealthy. So,
yeah, I do say I want to tell you about one of our sponsors, and at the end we thank this sponsor for support. So I think it's framed pretty appropriately.
I would agree.
I don't think anyone's going to end up in the middle of an ad. I don't think that's the way people listen to podcasts. So I appreciate your thoughts, Tarwin, but I don't know. I think it's okay. All right, we are sad to announce the death of Roger Sien, who was 64 years old, a UC San Diego professor, well known for his work with fluorescent proteins, and in fact, in 2008, got a Nobel Prize for his work on those. And I met him years ago at a Searle Scholar meeting when he was very young. We're both in our 40s, I guess. Nice fellow. And boy, is that young. 64.
Yeah.
Too bad. Well, I'm sorry to hear that. Lots of tweets about it over on Twitter. We have some Zika news. Anyone know what the latest Florida count is?
No.
Because the CDC website is not.
It's never up to date.
It's not current.
It says 35, which is not right.
But I know that there's now a travel warning to Singapore.
That's right. There's a travel warning to Singapore. Zika virus has now been identified in...
Well, there's a travel warning to Florida because it's a hurricane.
That's true.
Well, that's interesting thought, Dickson. Is that going to push mosquitoes around?
You bet it will.
Anyway, they have found Zika virus in mosquitoes in Miami Beach for the first time. They've been looking for these for a while, and they have 3 positive samples in a 1.5 square mile area where there have been locally acquired cases of Zika virus infection.
So they found those in the adults, or I see a picture on your computer of larvae.
Yeah, this is this is a control worker pointing out larvae.
Did they find them in larvae or in the adults?
Mosquitoes is what they say.
It's very important
Let's
to know
look
the difference.
Through this article. Yes, sir. Why is it important, by the way? Tell me.
Because of one of the papers that we're going to be discussing.
We'll talk about that. I don't know. It doesn't say here. I'm sure we could find it. It says 2,000.
I'm sorry, I just had an autoplay video come up with one of these stories, so be careful.
2,470 mosquito samples consisting of over 40,000 mosquitoes.
Which ones, though?
Well, they don't say.
That's not...
I suspect if they were going to be talking about larvae that they would have said larvae.
Maybe.
Yeah.
But maybe I'm just too
because
naive.
it's easy, much easier.
They do quote in this article 45 homegrown cases in Florida.
Okay, so that's our number for this week.
Most mosquito surveys are done by collecting the larvae, not the adults.
Okay,
Easier
well.
to catch.
Yeah.
Is that right?
Yep. And you're right, the hurricane is going to definitely raise havoc with...
Now, Scott Weaver is quoted in this article. He's a virologist. He's in the University of Texas Medical Branch, Galveston. He says the good news is the weekly number of cases isn't changing much.
Right.
If we were first seeing 5 a week, then 10, then 20, and 100, we'd be concerned. That's something we've been talking about here. There are not a lot of cases.
I'd like to also modify the last remark I just made, realizing that the hurricane didn't actually touch Miami at all.
Really?
No.
Right. It hit the Georgia. It's over a little. It's further to the west.
It's in the northern panhandle part. So it's
through the Gulf.
It might be that it didn't rearrange them. It might be.
Now, let me ask you, Dickson,
Yeah.
as an expert on mosquitoes.
Well, of the four of us here, I think Alan probably qualifies too, because your grandfather was an entomologist.
I know a bit about mosquitoes.
Yes,
I've studied it.
All right, so Dickson and Alan.
Right. Please.
We have 3 positive samples out of 40,000 mosquitoes.
Yeah.
Is that not a lot? Was that what you would expect? If it were dengue, would you have that similar number? West Nile?
West Nile is much higher. When it's endemic, you can find lots of mosquitoes positive for that.
Yeah.
And many species, but in this case, it's a monospecific transmission species of mosquito. So I would love to know if those were larvae or not, because that would indicate some vertical transmission.
Which we're going to talk about in a moment, too.
Exactly.
Now, they are
Alan, you agree with that, or?
I think this seems consistent with just sporadic cases. It's not.
Yeah.
I do not know the numbers though. So for dengue, so that would be the relevant comparison, and I would want to see or an even better comparison might be mosquito surveys from someplace like Puerto Rico,
exact,
or northeastern Brazil, where you really you have endemic spread going on.
That's right. And we know that dengue is not vertically transmitted.
Yes.
So all of those are mosquito to people.
Right. And I'm going to assume that this was adults that they were looking at, because we don't have, until very recently, we don't have evidence for this being a reliably vertically transmitted thing. And if you were setting up this survey,
yeah, it's true. That's a good point.
Back in May, you would go after the adults. So it's probably something like a gravid female mosquito trap, where you have a pan of water with a fan over it, and the females go into the water, go to lay their eggs in the water, and they get sucked up by the fan and trap them that way.
I wonder if Microsoft is deploying its drone-delivered traps.
Right.
Yeah.
Good question.
They are going to do aerial spraying in Miami Beach via helicopter, because they have a lot of tall buildings and they can't just walk around. So what do you think of that, Dickson and Alan?
Well, that didn't work here for the West Nile virus, so I don't think it's going to work.
Is that right?
Yeah, I think
sounds like a fun job.
You should attack the larvae in the breeding sites and try to get rid of them. That's the best possible way to do this.
It is best to go after the larvae in the breeding sites, but aerial spraying is probably a good idea too, because it's a large city. You're not going to be able to get everybody to clean out their gutters. You're going to want to have some additional insurance there.
You're going to get 1 day's worth of hatching. As soon as that stuff is destroyed by sunlight, which I hope they're using the photosensitive insecticide, the malathion, the next day to get another hatch, you've got to spray every day for how many days?
Right.
Mm-hmm. That's the big difficulty.
Now, our other story is CDC. Tom Frieden says the cupboard is bare. He has almost run out of the money that he got to fight Zika. He had gotten $222 million just for the CDC. He spent $194 million, and he says if we don't get more money, we're going to run out. And this is a political issue now.
What is he spending the money on? Do we know?
Well, when in Miami in particular, he's helping.
Who's doing these surveys
and looking for virus and diagnostics and so forth. And I'm sure they're doing a little research there.
Control
serology.
Or just surveys?
I don't know if they're doing controlled. Would they be participating in the spraying? I mean, that's a Florida Mosquito Commission issue, right?
That's right.
It doesn't say in this article in the Times. But the Congress is scheduled to vote on a $1.1 billion Zika package next Tuesday when they come back from vacation. However, Democrats have been blocking the bill. It's a Republican package because it would exclude Planned Parenthood from the list of providers that get new funding for contraception to combat spread of the virus.
That's crap.
Isn't it ridiculous?
That's crap.
Yep.
That's
just ridiculous. Mm-hmm. Yeah, Planned Parenthood really gets killed by the Republicans. Ooh. And there has not been any.
And this would be, what they're blocking is funding specifically for contraception. And if it's contraception to combat spread of the virus, we're talking about condoms.
Yeah.
So what no, go ahead.
Yeah. So it's just, you could earmark the funding for that, and it does. It's just ridiculous. It's just grandstanding.
So, Alan, I'm surprised you haven't said this, but I guess I will. So the headline reads, Republicans abort Planned Parenthood budget.
Yes.
Back on the comments about how effective spraying is, it was in this article that it was described, and I was trying to find it when you guys were talking about it. Aerial spraying has been highly effective, Dr. Frieden said, and the success comes from combining sprays that kill adults with sprays that kill larvae.
Larvicides.
Okay.
So the outcome goes against conventional wisdom that aerial spraying does not work against the Aedes aegypti mosquito, the type that carries Zika. And then they do say, for instance, that Puerto Rico, with the largest number of cases, more than 8,700, has no mosquito control agency, and the CDC had requested funding to start building one. Some funds were included in that $222 million, but not nearly enough. They weren't able to get a good running start on the mosquito control agency for Puerto Rico.
Okay.
Great. Thank you. No Zika infections in Brazil during the Olympics among athletes or visitors, according to World Health Organization.
In accordance with the CDC model we discussed.
Yeah.
By the way, it makes you wonder about the recent relaxing of regulations for travel to Cuba. And JetBlue had their first flight about 3 or 4 days ago.
Really?
It was announced.
Right.
Yeah, so they're gonna get up to 20 flights a day, commercial flights to Cuba, and so I wonder what the Zika situation is there.
Cuba already had trade with everybody else except us.
Right. But now you can fly from Miami, you see. So
yeah, now you can fly from Miami, but you could previously. I'm pretty sure you could previously fly from
Puerto Rico,
other parts from Latin America. You could not fly from Puerto Rico, but
Mexico,
but other parts of Latin America that are likely affected by this. So if it's going to get there, it's already had a chance.
CDC issued a travel advisory to Cuba, Dickson. Zika virus is being transmitted in Cuba.
It is, okay.
There you go.
Certainly is, yes. All right.
Dude.
All right, there is a Zika paper which came out this past week, which I thought we could do it relatively quickly. It was published in the American Journal of Tropical Medicine and Hygiene. Lovely title, isn't it? Yeah. It just like harkens back to the old days.
I'm a card-carrying member.
Hygiene.
That's right.
Well, this journal harkens back to the old days, doesn't it?
It does.
Yeah, and this is...
It used to be called the American Journal of Hygiene.
Vertical transmission of Zika virus in Aedes aegypti mosquitoes from the University of Texas Medical Branch in Galveston, Texas. And the first author is Saravanan Thangamani, Huang Hart Guzman, and from the lab of Robert Tesh.
I know him. He's a nice man.
How do you know him, Dickson?
From my years of going to those meetings and meeting with all those...
mosquito people?
They're entomologists. No, actually, he's more than that. He's established colonies of black flies in the laboratory, too. So he's a clever entomologist. He's very, very good.
All right. Now, the question here is whether Aedes aegypti and Aedes albopictus can vertically transmit Zika virus.
Right.
In other words...
And that doesn't mean coming down from the sky.
No.
Can the mother...
Dive bombing.
Can the virus get into the eggs that the mother lays... in the mother, so that she lays eggs with virus, and then they hatch, and those babies have virus. Now, why is that important, Dickson?
Well, because you can spread this in 2 different directions all at the same time, and plus it eliminates seasonality, basically. The mosquitoes themselves can continue to harbor this virus until such time as, let's say, people move back to an area, if they migrate from one place to another, that sort of thing. So it really complicates the epidemiology.
And it also complicates the mosquito control if your aim is controlling the virus, because the eggs are much more durable than the adults.
No question about it. And so we have one example of this now with West Nile in the United States, where in the northern part of the states, the mosquitoes can actually winter over, and they can harbor the virus, and they can give it to their offspring in the spring without taking a blood meal. Get this. They can make eggs without taking a blood meal. And that starts their cycle very, very soon after the spring thaw. So that's an
Which
unusual.
Mosquitoes are these?
Those are culex, and these are that's a very unusual pattern.
Why can they make eggs without a blood meal?
That's a good question. They've got a special biological clock that ticks away all winter long, and allows them to produce a clutch of eggs. Just
but
that
then
one time.
Other times, they have to get a blood meal. They have to get a
blood meal after that.
So West Nile is vertically transmitted?
Absolutely.
You said dengue is not, right?
It's been proven that it's not.
What about yellow fever virus?
No, it's not.
How about Japanese encephalitis virus?
No, it's not.
Wow.
What about chikungunya?
Chikungunya?
No, it's not. But this paper will supply the same kind of data that they got when they asked that question for those other viruses, particularly Japanese beetles.
Actually, it's also all that's in a table here.
Exactly right.
I don't know why I'm asking you. Well, they got all the viruses, the dengues.
Because I did read it, actually.
Well, actually, go ahead, Alan.
They kind of argue that those other viruses are maybe vertically transmitted. It's just not reliably.
No, but there's no evidence that they've ever recovered virus from eggs or larvae in the field.
I mean, in nature. These are laboratory.
In the field. Yeah. So you have to say in the field. All right. So then, therefore, it's probably not an important deal. But this is a laboratory-based experiment.
So basically,
they spill quite a bit of ink in the discussion. By the way, this paper is open access and quite readable. They spill quite a bit of ink in the discussion talking about the implications or non-implications of this.
That's right.
So basically, the experimental format, they take 100 female mosquitoes of each species, inoculate them intrathoracically with Zika virus, 10 to the 6 PFU per mil, and then they let them take a blood meal. They lay eggs.
Yes.
They collect the eggs. They allow them. Do they allow them to hatch? Yes.
Sure.
And then they look for virus in them, and they actually make an extract from them and inoculate it into cells, and then they do immunofluorescence to score for infected cells.
Yep.
Right?
Yep.
Mm-hmm. So, they did 69 pools consisting of 1,738 F1 adult Aedes aegypti. 6 of the 69 pools were positive by immunofluorescence.
6.
There were way more F1 adults than that. It's just 6, at least the way I read it, 6 consisting of that had 1,738 adults.
You're like 40,
because that's where you get the 1 in 290.
40,000, is that right? That number I remember for some reason. Anyway,
it was a similar number that they got when they did the dengue experiment.
And none of the Aedes albopictus pools with 803 adults were positive.
Exactly.
0.
Exactly.
So they have a table summarizing that, which they call the filial infection rate.
Right.
So for Zika, it's 1 in 290 in Aedes aegypti, and less than 1 in 803 in Aedes albopictus. But if you look at some of these other viruses, let's look at dengue. Aedes aegypti is not very good, but Aedes albopictus 1 in 200, so that's in the same ballpark as Zika, right?
Exactly. Right.
Mm-hmm.
West Nile, culicines 1 to 300 or 1 to 800. Aedes aegypti 1 to 60. Wow, to 1 to 70. That's pretty high. Japanese encephalitis can also be 1 to 80 in Aedes togoi.
And these are laboratory findings.
They are, exactly.
Right. And they put a big, as Alan said, big question. Does vertical transmission occur in nature? Answering this question is a much more difficult problem.
Yep.
Because you'd have to collect immature forms, larvae, and pupae.
Hence my question earlier. And
right.
We really have to get that other survey to tell us what those mosquitoes were. But
Dickson, has this been shown for any virus that in nature there's vertical transmission?
Yeah, West Nile virus.
West Nile for sure. Any others?
Not in mosquitoes, but there's vertical transmission in ticks for other infectious agents, of course, the Rickettsia, et cetera.
Now, in the article we just talked about before, the mosquitoes positive for Zika, they show a fellow holding larvae in a cup, right?
Yeah, they do.
Now, so you could look at them for virus, right?
You could.
How do you collect those?
Yeah, but that's another thing.
He's got this cup, which is very dirty, and he's putting his finger in it.
Yeah, but I'm going to tell you right now that that's a fake photograph, because that's not how you would collect the water from Aedes aegypti breeders, because they're tree hole mosquitoes, and they're in temporary bodies of water. So you'd actually collect the can, or you would, you could dip into an old tire. Sure, I guess you could do that.
Okay, so you have to do a lot of legwork, basically.
You have to do a tremendous amount of legwork. And look for them in all the peridomestic places that you have standing water.
Would you like to go do it, Dickson?
I actually did that
with
once.
you.
I've done that. I've done it. It's hard work. It is hard work. You're out all day long, and you're looking in places that you would never.
Yeah,
you're not comfortable looking in some of the places that you have to go to.
Well, Dickson, next time you go fishing, can't you go collect some mosquito larvae?
How would I do that, Vincent?
Well, you just told me.
Don't fly fishermen always collect larvae to see what stage of the hatch is?
Yeah, we do, but that's cold.
You showed me some scuds this week. Dick.
Right.
I showed you some scuds, and they weren't missiles. I showed you some.
Dixon brought back pictures of scuds from his last fishing trip.
Well, those.
What are scuds?
They're crustaceans that live in limestone streams.
Oh, are they like crayfish?
No. They're like amphipods.
Ah.
In fact, they're related to amphipods.
Do the fish eat them, Dickson?
Oh, they love them.
Interesting.
They're the shrimp of the freshwater.
I bet they have viruses in them.
Oh, of course they do.
Of course.
Everything has viruses.
But it's a great name. What is the other one called? There's another picture you brought back. Not a scud, but...
Brought a picture of bluets.
No, not flowers.
I brought some stonefly shucks and some isoniches.
Shucks.
Yeah, the hatched insects. I brought you the remnants.
And isoniches, yeah.
And I got you a live picture of one of those, too.
Bluets.
Good stuff. Bluets. No, I like to pick pictures of flowers.
Anything else on Zika before we move on?
Only that Singapore is now under red alert.
Oh, there was actually in this paper there was an interesting mention of the issue that there are other flaviviruses that are mosquito specific that could confound this.
Ah, yes.
So they point out that there's accumulating evidence for mosquito specific flaviviruses that don't seem to affect humans.
Sure.
And they raise the possibility that there could be superinfection exclusion
it's very
of these viruses. So that, and since these mosquito-specific flavies are presumably in a lot of the lab cultures, that could explain some of the inconsistent results that people get from that.
Interesting.
Kathy, in Europe, are they talking about Zika a lot?
I didn't hear any discussion of it.
You were at an ADNO meeting, though.
Yeah, that's true, too. But, yeah, and I didn't bring it up. So.
Right.
All right, I'd like to tell you about our sponsor of today's show, Curiosity Stream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles, 600 hours of content, founded by John Hendricks of Discovery Channel. And as you know, Discovery Channel has nonfiction content, so that's what you'll get at Curiosity Stream, nonfiction, not reality TV. You can get it on the web. I mean you can watch the content on the web, or you have one of these devices like an Apple TV, an Amazon Fire, or Kindle, a Roku, Android, iOS, or Chromecast. You can get it on those as well. It's available in 196 countries. They have a wide variety of science, technology, nature, history, interviews, lectures, a lot of cool stuff. For example, Stephen Hawking's Universe, a series where Stephen Hawking traces the history of astronomical theories and technology. Deep Time History, a 3-part series on the 14 billion year history of the universe. Some of these are one piece, some of them are multiple, some of them are 20 or more. It really varies. Underwater Wonders of the National Parks is the 100th anniversary of the National Park Service in the U.S. They take you underneath the bodies of waters within the parks. And they have one of the largest nonfiction 4K libraries around. That's ultra high definition. So you can see those shucks and chuds, or scuds and shucks, whatever. Sorry to bring you in on an ad, Dickson.
It's okay.
This is still an ad. They have monthly and annual plans available. They start at $2.99 a month, less than a cup of coffee, or a bottle of water here in New York City, in fact.
Right.
And that could be one title on a competing platform. Check out curiositystream.com/microbe and use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series. Completely free for the first 60 days. That's 2 months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. And, go visit it. Even if you don't intend to check it out or subscribe, just go visit. So they see lots of people come from TWiV and click on the site, and they'll go, oh, we should continue advertising there. That works. Mm-hmm. We thank Curiosity Stream for their support of TWiV. All right, our paper today is uber cool. and that has nothing to do with the car company. It's an article in Nature just published called Uncovering Earth's Virome.
It's a nice, modest
and this
title.
Is going to be covered for us by Earth's virologist who can explain it.
That's right.
Where's that? Who's he?
Yeah.
Earth's virology. I'm actually the virology professor because probably some people would take issue to me being an Earth's virologist, but I am an Earth. I'd like to be Earth's virology professor, and this is by it's out of the Joint Genome Institute in the Department of Energy in Walnut Creek, California, and the Lawrence Berkeley National Laboratory, and a company called Metabiota in San Francisco. And as you'll see, it's all computational biology. Very cool. And the first author is David Paez Espino, and the other authors are Elo Fedrosh, Pavlopoulos, Thomas Hunterman, Mikhailova, Ruben Ivanova, and Nikos Kirpides is the last author. And as everyone knows, viruses are the most abundant, listen, this is a quote, biological entities on Earth. They call them entities. They're not sure what
that's a good
they
word.
didn't want to incur the wrath of TWiV by calling them organisms.
Or Dickson, microbes, that's right.
They're not microbes.
But, finding all of them is hard. So what they do here to try and fill in these gaps and also to find out where they are and what organisms they infect, they simply examine all of the metagenomic data that has been generated and deposited on a computer.
Wow.
Right? And they just use new ways to look at it, and they end up uncovering scads of new viruses and viral genomes, and they even make some associations with hosts, as you will see. So they explored over 3,000 metagenomic samples. So these are samples that people have done total high throughput sequencing on. They come not just from people, but from the ocean, from lakes and dirt, and what they call engineered sites like sewage treatment plants, and what's another engineered site?
Buildings. The whole built environment.
Water purification?
Yeah, water purification. Yep. Thank you. That's the other one I was thinking of.
Okay.
And this is like, how many terabytes of
a lot.
data?
I read that.
5 times 10 to the 12th, or so 5 terabytes.
5 terabytes of sequence data. They end up uncovering 2.1 gigabases of sequence data. Viral sequence. They increase the known viral sequence space by an order of magnitude. Really, it was so cool. I don't understand a lot of the computational approaches, but the first thing they do is, so previously people say what proteins are exclusive to viruses, and then they use that sequence to explore sequences, and they said, here, this is not good enough. So what they did is they took all the known viral protein families of isolated viruses. They took all the proteins of them and used that as a database to search the sequence, this 5 terabytes of sequence space. Okay, so they have what is this called? Isolate viruses. These are IVGs. These are viruses that have been isolated in sequence. They take all the proteins from them, and then they take this metagenomic viral contigs, and they use the proteins to develop that. So the metagenomic viral contigs are what they identify in the in these samples that have been published already there. So
they take the viruses that we know are viruses that have been sequenced. They use that as bait to pull out everything that looks like those to try to get a good chunk of the rest of the viruses that are out there.
So as I said, they looked at 5 terabytes of sequence. That's 3,042 metagenomes. So there's a site where you can access these, and they're, as I said, geographically and ecologically diverse. So they were able to identify 125,842 putative DNA metagenomic viral contigs, which increases the known viral sequence by 17-fold, the number of viral genes by 16-fold. And here's the number that's really amazing. These sequences encode more than 2.79 million proteins, 75% of which have no sequence similarity to proteins from known viruses that have been isolated. 75%, brand new. Doesn't that make you chill? it sends chills up my spine that there's so much out there that we don't know. It's just amazing. And that we've talked about that before, right? Like 90% of this sequence is unknown and so forth. That is just amazing.
Vincent, I have a great book for you to read then if you're thrilled with stuff like that. And it's called Diversity of Life by E.O. Wilson.
He wasn't even close.
No, he doesn't have to be close. All he has to do is say, we don't know anything. And here's why we don't.
I know we don't know anything, but when you put a hard number like that on it, it's really impressive.
Well, and this is bear in mind, this is from sequences from samples, from things people have thought to sample.
Right.
Yeah.
And it's from samples that are DNA based.
Yes.
So this is not telling you anything about RNA viruses.
That's right. It's all DNA-based.
So does Craig Venter's data fit into this, or does he keep that to himself?
Yeah, the Ocean Global data set is part of this.
Good.
Yeah, he submitted those to the database, and in fact, I guess we're going to talk about this in a moment, but there's a very clear bias in the total set toward obviously human samples and marine samples.
Okay.
Because those are the things that people have sequenced most of. Sure. But people find entirely new animal species from time to time, and nobody's taken one of those apart and sequenced the viral or the metagenome that's in its gut. So you can multiply out this is just what we don't. This is just the known unknowns that we're talking about.
The cool thing, remember, keep in mind, is that this is sequence that's already been done. It's sitting in a database, and it wasn't mined appropriately, and just by going through it, they've discovered new viruses.
Well, it was mined appropriately for whatever somebody was looking for, and then they moved on. But there's more there, and nobody picked up a pipette in this whole paper.
And remember, also the other point is that none of these sequence sets have virus enriched. Like a lot of the studies we've talked about, they try and enrich for particles and just sequence that. These are just total sequences.
Right.
I think somebody did a little bit of pipetting because they did mention a little bit of particle stuff somewhere.
Did they? Okay.
Yep.
So would any of these results allow any further investigators to go to places in specific to try to mine out the whole family of viromes that might be there?
Sure. Now you have these contigs these are viruses, and you can take that sequence and go out and look for similar things.
So does each sample have a GPS?
Yeah, it does.
Yes.
Okay. In fact, there's a cool map. Yeah.
Excellent.
That'll be so. The highest covered category were marine and human associated habitats, where there are more isolate viruses available. And there's a low coverage category, includes soil, plant associated, engineered samples, and they say we need to look there more to fill that in. The length of these these contigs range from 5 kb to nearly 600 kilobases, and they can see the circular ones based on the N sequences. Average size of the circular contigs, 53,600 base pairs, and they got the largest phage recovered to date, a 596 kilobase genome from a bioreactor sample. So lots of phages in these samples, lots and lots of phages.
Yes.
Then they group them. They call these quasi-species grouping. Now, it has nothing to do with a quasi-species, but they're trying to put them in species. That's why they're quasi, because they don't really know. They're doing it computationally, and they were able to put together 18,470 viral groups on this analysis with between 2 and 365 members in each group. But the majority of these groups, 95.9%, didn't have any isolate viruses in them. So they put together groups by homology that would have similarities, and sometimes there was an isolate virus, one of these known viruses that we talked about, but most of the time there was not. So that just, again, emphasizes how much new stuff there is here. And then they want to know what these infect, right? That's really important to know. So for the for the groups containing isolate viruses, they know what those infect. So they say we projected that onto the other viruses that we've discovered, assuming that they infect the same host. But the cool thing they did is use CRISPR-Cas sequences from prokaryotic genomes. And these, of course, CRISPR-Cas contains spacers that are derived from infecting viruses. So if you have the genome sequence of a prokaryote, you can look at it and match it to a virus.
How about that?
So they had 3.5 million spacers from prokaryotic genomes and they matched it up with their sequences and were able to say which viruses.
So they interrogated the bacteria.
They interrogated the bacteria.
That harkened back to a nice.
Have you seen a virus like this?
Yes. Right. Nice.
And then they also used, some viruses have tRNA genes in their genomes, and they're thought to have come from the host, and they use those as well. They were able to identify 9,992 putative host virus associations, but that's only 7.7% of their contigs that they identified anyway. Most of these previously unknown, and include hosts from 16 prokaryotic phyla for which no viruses were previously identified. So, we always say, is all life on Earth infectable? Probably, but we haven't found a lot of it. Well, they just filled in a lot of it here. Right. It's very cool. Now, the other cool aspect of this host association is they say most of they find a lot of viruses with a narrow host range that just infect one or a few different species, but there are some exceptions of examples of viruses that have very broad host range within multiple genera, multiple hosts from higher taxa, including different phyla. So that's very cool. That I think that's really interesting that you can have some of these broad host range viruses. Now, what about
vector-borne viruses?
Absolutely.
You could think of as broad host range in insects and mammals.
Absolutely.
Then they look at the biogeography of these viruses. Where are they from? And of course, a lot of the sequences are from the oceans, and they find that there are very distinct patterns of viral location based on water column depth and distance from the shore. Right. So they have these very nice charts where they're showing the different isolates in the water column, right? And they use a term bathypelagic.
Yep.
So this is at a certain depth of a water column, right?
It's in the deep ocean.
Deep ocean is about 1,000 meters down.
Right.
No light penetrates. It's extremely cold, completely dark.
Right.
About 1% of all ocean species live in this zone. Sperm whale can dive this deep and might find giant squid, anglerfish, gulper eel, or snake dragonfish.
Got it.
I don't want to go there. That sounds scary, doesn't it?
A little bit.
Anyway, they say bathypelagic samples from different oceanic basins have very similar viral profiles.
Oh, interesting.
Isn't that cool? But they say that's consistent with the fact that the microbial communities are pretty uniform.
Not only that, though, the Earth has a certain circulation of oceans, which would easily distribute those.
Yeah.
Right, but they found more variation in the epipelagic zone, I think.
Yeah, but
up in the higher part of the water column.
Surface,
yeah,
because you're going to have more temperature variation there.
Yeah. That's true.
But the currents are different at different depths, and it's probably just that by the time you get down to the bathypelagic zone, there are so few things that can live there that your viruses are going to be associated with those few things all over the world.
Mm-hmm.
And by the way, there are two layers under that, the abyssopelagic and the hadopelagic. So I guess the hadopelagic is the ocean of hell.
Now, just to take a little diversion, Dickson, you show me a picture of all the water on Earth as a little ball over
that's
the
right.
It's like, this is really deep. This is 4,000 meters deep.
I
How
know.
could there be so little water?
The Earth is 25,000 miles in diameter.
All right, it's just a little bit. Then they looked at the distribution in human...
Circumference. I meant circumference.
Human microbiome samples. They show clear body site specificity, so they have samples from different parts of the body, with only a few groups of viruses in fecal and oral samples. And what they find many sequences shared between samples from the same body site of different people.
Right.
More than 30% of intestinal and 50% of oral viral sequences were shared by at least 10% of sampled subjects.
How about that?
So people are fooling around more than we know about.
Is that what has been found for bacteria? For instance, in microbiomes or...
Yeah, there are certainly shared species.
Yeah,
right?
I guess I had the feeling that, I guess that over time, one individual is more similar to themselves than to even family members, I think. So that's what I'm thinking of. Okay.
The other cool number here, they estimated the amount of viral sequences in 550 fecal and oral samples. All right. Is the fraction of sequences that are viral. It varies from 0.2 to 54% of the total sequence.
0.2 to 54, that's a big range.
That's a range.
It's a big range, but in some samples, half of the sequence is viral. Right? And that's higher than previously reported.
Wow.
I think that was pretty cool.
But the average is closer to 3.4 to 7.4 in oral and stool.
Yeah. They also could see a lot of a number of sequences they found in multiple sites, and they think some of these are contaminants, human and lab contaminants like phage and lambda phages that we use as vectors, sequencing and molecular weight standards, and also phages of Propionibacterium acnes, which are common inhabitants of our skin. But there are still viruses found in diverse environments that are not contaminants or any of these things. So again, you can find them in aquatic samples with big differences in salinity, like freshwater and hypersaline lakes, oil-contaminated wastewater, and human and oral fecal samples. So these different environments would have similar viral populations.
Did they look in any regions that we would consider to be strictly archaebacteria inhabited, like hot springs and...
Well, I think those are there are.
I think there must be
in here.
included.
Yeah, they're included in these.
Those are in there.
They have hydrothermal vents. They have oceanic sediments, coastal waters, coastal sediments. Estuaries.
Did they look in rock? Solid rock?
I don't know if they looked in rock. No.
Well, it's what other people looked in.
Yeah, but
so if anybody has sequenced rock and done a metagenomic,
no, they have. They actually have.
Then that would have been in the database.
A big study that went down into the gold mines in South Africa and worked on the snot that comes out of the rock.
So they say there's a limited number of ubiquitous virus, cosmopolitan viruses, they call them. All right. The last thing is they generate a global map of viral dispersal. And they to link the sequences with geographic coordinates because they have the GPS, as you said.
Yes.
And many viruses have similar niches across large distances. The most prominent is the marine samples, which you can look at the marine map. You see similar viral sequences all over the globe, right?
Yes.
It's really cool. They have these blue dots that show you where the samples were taken and then a line connecting it to another sample that is very similar. But they also, and they say this is consistent with transport of viruses along ocean currents, right?
Yeah.
Sure.
Makes sense.
Then they also see some connections between non-marine viral groups like fresh waters across continents, thermal springs, terrestrial soil samples, etc., which is the bottom graph. Is this open access, Alan? Probably not.
No,
right?
Unfortunately, it's not.
This is a beautiful picture.
It is.
With all these connections. Now, one picture here that I like is a picture of a human where they've sampled viruses at different body sites, like intestines, urogenital tract, mouth. And then they showed that there are similar sequences to, I assume, viruses in the ocean, that's the globe, engineered viruses from plants, and viruses from animals, right?
Engineered meaning engineered environments, not viruses that were created in the lab.
So for the engineered, there are 311 contigs that are very similar between what you get in humans and an engineered, I guess a sewage treatment plant. That would make sense,
well,
right?
Yeah,
that would be, yes.
That's still human.
And but 984 shared contigs or sequences. I'm not sure what the number is between
and
humans
engineered
and
environments are mostly inhabited by people.
Yeah,
indeed.
But that's low. It's the animal connection is higher, almost 1,000.
It's higher. Yes.
So I guess we eat, animals, right? And we get lots of their viruses that way.
Yeah.
Especially in our intestines.
And we are animals.
We are animals. Anyway, that is an overview of what I think is a really cool paper.
And the distribution, the marine samples that have common seem to be in common. Those are easy to explain with ocean currents. They seemed at a loss to explain things like the freshwater viruses that are similar in different places, or the soil viruses. But I can think of at least two explanations for that. One is air currents, because we know huge amounts of soil get transported around the world on the jet stream, and the other is the continents used to all be attached.
It's true.
So it's entirely possible that there are, especially phages and such ecosystems that that are now in the Great Lakes that used to be in some lakes in Gondwana land that got split up, and now we have them spread here.
And you should also consider birds, migratory birds.
Yeah.
Migratory
Plus,
birds.
the air is full of microbes, tons and tons of fungal spores, for example, right, which are floating on the air and going all over the place. So that could do it as well. Very cool.
I just wanted to say that I spent a lot of time trying to understand this, and I found the abstract and the introduction and the discussion the most accessible. And I wanted to highlight a point from the discussion that relates to what I was trying to say before. They say it's more surprising was the significant human virome sharing between unrelated individuals and the identification of viral quasi-species distributed across markedly different habitats. So that virome sharing between unrelated individuals was kind of surprising.
Right. Now, where did you see that they'd picked up a pipette?
They didn't say a pipette, but they said something about particles.
Right. They were mentioning that genome size generally correlates with particle size.
No. It was more than that.
Okay.
I'll find it. You guys can move on. I'll find it. Yeah.
I mean, it's fine that
I kind of read through. I read through a lot of this. I mean, almost all of it, and still.
It'd be cool if there were no experiments, though, because there's so much. There's so much here.
There's so much material here. Yeah.
Yeah, but the materials and methods, I really tried to make sense of, and
Right, and they did do experiments. These are experiments that they, you build these searches and these algorithms, and there's a hypothesis, and you test it, and I'm sure there are quite a few failures of the experiments that we're not seeing here. So this is a heck of a lot of work. I am in no way demeaning it by saying they didn't pick up a pipette. I'm just pointing out how far research has come, that you can do these types of analyses. And that we need to do these types of analyses because the data sets are so massive that no one group can analyze them.
Yep. All right, let me tell you about our other sponsor of this episode. Okay, Dickson?
Go for it.
Are you leaving?
No, why would I do that?
Our second sponsor is Drobo.
I'm hanging on every word.
No, you're not, but that's fine. Drobo, a family of safe, expandable storage arrays for your computer. If you need a lot of storage space, you should get a Drobo. Drobo takes care of the problem that hard drives fail and that you fill them up. So these are multi-bay devices. You slide in as many drives as you need, 2 or 3 or 5 or 8 or 12, depending on which one you buy. And they're all fused together, not physically, but virtually in a single hard drive. And their data is redundant across every drive. So you can keep adding drives to get more storage. And if a drive fails, you just take it out and put a new one in, and all the data are replaced. And there are all kinds of different models that are available. I think they have 8 different models, 8 products, and depends on what you want to do. Now, the cool thing, which I want to tell you about this week, is that a number of years ago, they told developers, go ahead and develop apps for the Drobo to expand its utility. Because right now, you could put files on it, but maybe you could do much more. And these are called Drobo apps. There are 4 different kinds. For example, a number of companies have made apps to let you back up to the cloud. So you could back up your Drobo to the cloud, which is great because, if you have a Drobo with lots of files on it and your house burns down or it gets stolen. I heard once that Francis Coppola lost a whole bunch of scripts on a hard drive because people broke into his house and stole it, and he didn't have it backed up. Scripts for movies. So put it in the cloud. So they make cloud backup apps. If you want to develop a website, they developed apps so you could use your Drobo to serve up a website. Put all the right software on it and so forth. That's very cool. Some of you may have heard of Plex.
So your Drobo could be the cloud.
Could be the cloud as well. That's right. But make sure you back it up anyway.
Yes.
if you've heard of Plex, it's a piece of software that you can use to stream media from your computers to your TV or other devices. And they've got apps to let you do it from your Drobo. They've got apps to make your Drobo work like a BitTorrent client to gather or distribute files from the internet, or to even use it as an email server. Really cool stuff. Lots of cool apps which you can get from third parties. And Models 5N and 810N are network storage systems, so you can extend these capabilities with Drobo apps. And again, these are free applications installed from the Drobo dashboard, which are all being supported by Drobo. So you plug in a network, what is it called? A network storage system. You take your Drobo and simply plug it into your switch or router, and it's now available not only on your local network, but if you use some of these apps in many other places as well. I think that's really cool, really expands the use of a Drobo over what we had originally. We talked earlier about how Drobo supported us years ago. This is before all of this. You could use it as a local storage solution, but now this is even greater. It's really cool. TWiV listeners can save $100 off on their purchase of a Drobo Mini, Drobo 5D, 5N, so 5N is the network-attached Drobo, or any 8 or 12 drive system. Go to drobostore.com and use the discount code MICROBE100. We thank Drobo for the support of TWiV. Cool stuff. I like Drobo. I use them, and I use them in the lab as well, and I think for scientists they're really cool as well as for everybody else. Let's do some email.
Wait.
Yes.
I want to just clarify.
Yep.
Okay, so it was the fact that although the vast majority of the data sets were generated from double-stranded DNA using an untargeted approach, 59 samples underwent particle enrichment, viral DNA enrichment, or library construction with sequencing protocols optimized for the recovery of viral sequences. But Alan is right. Those people who did that pipetting were the people that then put that stuff in the IMG/M database. Yeah.
Cool.
Okay.
Totally dry paper. No wet experiments. Although someone had to do the wet experiment. Yeah, the ocean.
Yes.
All right, email. Theo writes, "Hi, TWiVers. I think somebody mentioned John Oliver some time ago. He finally touched on vaccines. Well, a little bit. He sends a link to a YouTube video, which I think, fortunately, my computer is not attached to the mixer, so you guys won't hear it. I fixed that a long time ago. I think we've here. John Oliver discusses
like we've seen this.
how and why media outlets so often report untrue or incomplete information. I think we've seen it, but it's really good. A lot of people have sent it in. They always in Frankfurt. All right, Alan, can you take the next one? I don't want to read this whole thing.
Okay, so I'll do it. Yeah, I talk pretty fast. Bodan writes, "Dear Vincent and TWiVers, in TWiV 386, the dolphins did it. You asked the question whether modern humans carry unique endogenous retroviruses. I wonder if that's possible given human behavior and ecology. It may have been possible when humans lived in closer quarters in Africa with so-called sources of retroviral agents, but how would such retroviruses infect humans and not infect our more exposed primate relatives or lower orders first? Are we also not the ultimate and usually protected predators who no longer intermingle directly with a broad range of species as we once did?" Shall we address that and then move? No, let's do the whole thing. There are, however, currently individuals infected with HTLV 1, 2, 3, and 4. The last three appear to cause minimal disease. HTLV 3 and 4 are African retroviruses. They appear to be related to their simian counterparts. They may not yet be endogenous retroviruses, but they appear to be endemic in certain human populations around the world. Perhaps someday they will be able to enter the germline and thus become endogenous if they provide the host with a specific benefit. HTLV2, at least the sources that I've read, appears to be a New World virus, possibly brought to the Americas by migrants from Eurasia millennia ago. No clear primate relative has been identified according to my limited knowledge and review for this note. Perhaps not enough research has been done. It, too, is not endogenous. The same may hold true for HTLV1, but its presence in Japan, in addition to South America, the Caribbean, and Africa, links it perhaps to the Portuguese or the Dutch and the slave trade out of Africa. It, too, is not endogenous. It would seem to my research untrained mind that so-called non-disease-causing retroviruses would have a greater chance of hanging around their host longer and thus of eventually entering the human germline, especially if they provided some advantage to the host. At the very minimum, they should not alert the host's immune system. I would appreciate your collective insights. Then you've started a great learning resource. I'm using your podcast to educate myself about the nature of the virosphere and of the human virobiome interactions while I do research on a book about the incubation of HIV-1. I do have another question. How would one create a similar resource for doctors, something that would broaden their scientific horizons, help educate them, and enable them to rediscover the joy of taking care of patients? I would like to contribute a pick of the week in your honor. Shall we do that during picks?
Yeah.
Okay, we'll do the pick during
picks.
During picks.
Okay. Interesting bunch of things here.
And Bodan is an MD MPH.
Well, I don't know. I don't know the answer here. So, I mean, obviously, if a retrovirus kills you, it's hard to
pass it on
for it to endogenize, which means go into your germline, right? Now, I mean, I think we inherited a lot. Homo sapiens probably inherited a lot of endogenous retroviruses from ancestors.
Wait, unless you were dead by cannibalism.
Yeah.
Right.
Oh, did early humans do that?
Yes, they did.
Of course.
Yeah, that throws a monkey wrench in the system. That makes it complicated. But
a Darwin's monkey wrench?
Really an ape wrench.
An ape wrench, that's right. A non-human primate wrench.
Obviously, if I think, a retrovirus could still cause disease and be endogenized as long as people survive. It doesn't have to be mild, right?
Yeah, nothing kills everything. Right. So
there are always survivors. And with Zika, for example, even though it's not a retrovirus, only 20% of people get overt disease. Yeah. The others are infected. So, if you had a retrovirus which caused 20% serious disease, the others, it could endogenize. So I don't know that that's necessarily a requirement. Even not alerting the host's immune system, if the virus is long enough present before the immune system clears it, it might have a chance to endogenize, right? And you only need to get into one host's germ cell, and then, in 10 or 20 or 30,000 years, it's in most of the population, maybe.
That which does not kill us endogenizes.
That's right. That's right. I don't know. Does anyone have any more learned thoughts on that? How do you start something similar to TWiV for doctors? Is that right?
Yeah.
Yep.
I don't know if any doctors listen. By doctor, I mean someone whose primary job is to take care of patients on a daily basis. They are really busy.
Well, we got TWiP.
Yeah. And I don't know if anybody listens. Oh, that's right. On TWiP, we do have some docs. Yeah.
And we have case histories.
We get letters from practicing MDs on TWiV.
See, what we don't do on TWiV, which we do on TWiP, is that we present cases.
Yeah.
On TWiP. So we could do that on TWiV if we wanted.
We don't have a MD.
We could get one.
No, but he's asking how would one do this? Right?
I think an easy way would be to create a podcast and make sure it gets CME credit.
Yes.
Listening to it.
That would be perfect.
Which is not easy to do, but then you're going to get lots of listeners.
Absolutely right.
You would also get
what
lots
about
of
that Puscast guy?
And you'd get lots of money.
Sorry, Kathy, what?
What about that Puscast guy?
Puscast?
Yeah.
He's an MD, and I assume that must appeal to some physicians.
Pussy physicians, yes.
I don't know.
Yeah, Mark Crislip. Mark Crislip has a podcast called The Infectious Disease Puscast. He has quite a few things, actually. Check him out, Boden. Maybe you can collaborate with him. He does basically a weekly or bi-weekly literature review, and then he limits it to 30 minutes. So that's good also. Yep. But I'm not sure about the joy of taking care of patients. He's always complaining about patients, but you should check it out, Boden. But I think it's possible, yeah.
And probably, I would say maybe don't do it for CME credit.
Yeah.
First of all, that's a kind of a high hurdle to clear for a podcast that you're just getting started. But secondly, my own household experience has been that MDs treat CME as a chore to get through. It's not so much something they'd listen to during drive time.
Okay. All right. What's this, Kathy? You want to talk about this?
Oh, no, I pasted it down below with the pick. Is that what you're asking about?
That's from the ad. That's from the ad that's being discussed in Second Week for Boden.
Right, and Alan cut part of it, but I just copied.
Yes, you copied. I was about to copy the rest of that down, and you did.
Dickson, can you read Richards?
I will attempt to do so. It's only a sentence and a half.
Well, you could read more.
So here we go. Richard writes, how can an ERV acquire functionality such as the placental ERV? It becomes captured by an already successful system. How is its DNA expressed when it is a newcomer without molecular friends to enable it? Probably a dumb question, but I am not a scientist. Love your shows, Richard.
Not a dumb question.
No.
ERV, endogenous retrovirus, he wants to know, how can this be captured and be beneficial? Well, the key here, Richard, you say without molecular friends, but it has molecular friends. Because
that's how it got that's how it infected.
Yeah, and it's been brought in by cellular processes, and the viral DNA integrates into our genome, and then it's expressed by virtue of using cellular processes.
Right.
The virus doesn't do anything on its own. The mRNA is made from the endogenized DNA by the cell, and that gets exported out into the cytoplasm and translated into protein. So it has lots of friends. And the real question, I think, here is how would an endogenized viral gene end up being useful for people, right? So the placental protein syncytin is a good example of that. You know, at one time, years ago, these mammals, which are now having placentas and giving birth to live babies, they were laying eggs. They didn't have a placenta. So, at some point, an endogenous retrovirus went in, and this syncytin was made, and it started fusing cells in certain places. And the ones that could form this placenta somehow, it's hard to know the exact sequence of selections and evolutions that occurred, right? Now, you can imagine it gave some advantage to some animals to give live birth, and they survived. So, Dickson, do you think giving live birth is a selective advantage over laying eggs? I guess the eggs can get eaten by predators, right?
Yeah, but if you look at the success rate of dinosaurs, they were probably the most successful group of animals that's ever lived on this planet, and they were all egg layers, every one of them. And
if they were so successful, why aren't they here now?
You could say that.
Conditions changed.
You could say that about the aardvark too. So
they were really successful because they were dumb. And they didn't destroy the world.
They had lots.
No, they were successful because they succeeded in radiating into a variety of environments,
exactly.
And under the conditions at the time, it was a good body plan and a good strategy, and they filled a whole bunch of niches, and they were the dominant. What were they? A order?
Oh, they were at least several.
Yeah, the dominant classification of large organisms on the planet for hundreds of millions of years.
And they guarded their eggs. I mean, they have evidence that the mother stayed where she laid those eggs until they hatched, and then she encouraged them to go forth and multiply, as it were. And we have an equivalent to that today in the form of an alligator that has a similar behavior with regards to her eggs. And so it's possible.
I'm not sure it would have to be an advantage related to live birth versus eggs.
Yeah.
I agree. I don't know what the selection would be. Who knows?
Well, marsupials.
Yeah, there are plenty of egg-laying animals still around.
That's right.
It's not a bad idea.
No. And marsupials, by the way, have sort of a halfway house. They give birth to a tiny, 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 tiny little version that then suckles for 6 more months until it matures enough to become independent. So you've got some intermediate forms to look at in that sense.
I lately have been thinking a lot about trying to figure out, the selection for a certain property and how the sequence of events. It's really difficult.
It is.
Because you don't know all the details.
You don't have the intermediates.
Well, and in fact, the advantage of a placenta may have given us an advantage over the marsupials.
Mm-hmm.
Rather than an advantage over egg layers.
Yes. Yeah. That could be. That could be. Yep. Dickson, do you want to take the next one since it was short?
Yes. Robert writes, this is even shorter. No, it isn't, actually. He says, when I saw this CNN story, I thought, why is it necessary or a good idea to clone a virus? Zika, in this case. And then he gives a reference to the cloning of the virus. Maybe the idea is to have a reference virus, which is easy to modify. Then modifications can be made to the reference and large quantities of virus stock easily generated. Perhaps it could be a topic of discussion for an upcoming show. I've always enjoyed listening to the podcast. So
this is an article reporting the infectious DNA clone of Zika virus. So he wants to know why would you want to do this?
right.
And he pretty much hit it on the head. Have a reference virus which is easy to modify, and you can then make those and make large quantities of it.
Yeah.
So
the key
you want to make mutants. It's a genetic
It's an RNA
tool.
virus. It's an RNA virus, and RNA is very difficult to work with in the lab.
Right.
So whereas we've got all kinds of tools to work with DNA, so it turns out you can make a complementary DNA copy of the RNA virus, then do your tinkering on that DNA copy and turn it back into RNA in the cell and experiment, which makes it a whole lot easier to study the virus.
But now the listeners that are not virologists are going to ask you, Alan, why is RNA so much more difficult to work for in the lab than DNA? And you can say, because...
Well, we have restriction enzymes that cut DNA conveniently, and RNA keeps getting degraded by all the RNAs that people have spilled all over the lab from their mini preps.
Or just have on their skin.
Exactly.
Or just have on their skin. Yeah.
Exactly.
Or if they accidentally put it in sodium hydroxide.
Yeah.
Something that's even pH 8. Yeah.
Right.
There's a long history of infectious DNA clones of viruses, Bob.
Yes, there
way
is.
back to phages and other viruses. It's been done since the 1980s, actually late 70s.
Yeah.
So this is not new technology, but it's new for Zika, of course.
Right.
Kathy, can you take the next one?
This is by, I think, a different Bob. I wondered if the fusion peptide is a stable vaccine target or whether it is subject to mutation eventually making such a vaccine useless. And he sends a link to a news release from NIAID specifically, that describes that they've discovered a new HIV vaccine target, which is the fusion peptide of the HIV virus. And some ugly pop-up just came up. Okay, I got rid of that. Anyway, so Bob raises the question, is this fusion peptide going to mutate, and is that going to be a problem? And at the end of the press release, because he's probably thinking ahead to this fact that at the end of the press release, they say that they're now working to create a vaccine designed to elicit antibodies that will react against this peptide, this fusion peptide. Because backing up a little bit, what they found is that there are some people who have some antibodies to this peptide. People who've had HIV have antibodies to this peptide, and it could be a vaccine target. But just as an example, I tried to figure out how stable that fusion peptide is sequence-wise, and I found a paper indicating that some resistance to a small molecule inhibitor of HIV, the resistance arose specifically because of mutations in the fusion peptide.
Yeah.
So I think Bob is exactly right that a fusion peptide sequence is not necessarily stable, and so that could be a problem.
Yeah, there are antivirals that are actually short fusion peptides. They're called Fusion, and you get resistance.
Sounds like a Pokemon.
Very quickly. Speaking of Pokemon, I was walking on the boardwalk with my son several weeks ago.
Yeah.
And we passed this house, and we've always seen this house. They have a model of a dinosaur and a bull in the front yard. And all of a sudden, he was playing the Pokemon game. What is it called?
Pokemon Go.
Pokemon Go. And all of a sudden, a bull Pokemon showed up on his screen.
Really?
Now, did they really know there was a bull in the front yard of this house? I doubt it, but it's amazing. Maybe they saw it on the photos from the satellite, right?
I bet they did. Yeah.
Kathy, do you want to take another one, Kathy, since that was short?
Sure. This is another one from Bowden.
Yeah.
That one? Yep. Dear Vincent and fellow TWiVers. And he first attaches a link from the Washington Post that describes the Vaxxed movies. And he writes, "You guys and gals did not do justice to the film Vaxxed. The Washington Post did. I also believe that autism is a fascinating medical social problem and tragedy for the families involved. It is a very real illness, and neuroscience is as fascinating as immunology. Would it not be possible to hold a TWiV or other TWI on the real science of autism? Could we doctors and scientists admit to what we know and do not know about autism for the benefit of the patients and families involved? You made the mistake of considering this film a documentary. It appears to fall rather in the terrible horror film exploitation category. I will never trust your movie reviews again." And he puts a smiling face. Okay. "I think there is a danger in taking crap seriously. it lends it credence. The movie review approach works better in this case. Even a half a star is too much. There is nothing that is scientific in this film, so criticizing it on its scientific merits is not productive. Scientific was in quotes there. You gave this Trumpish film more justice than it is worth. Cheers. I love your podcasts, Bowden. And the article in the Washington Post just basically says that it's a really terrible movie and describes various aspects of it. I think it's just a different approach from what we did. So it's worth taking a look at.
They probably have more liberties than we do, too.
I think an autism episode is good. I'm not sure we're the ones to do it. Maybe there are some other podcasts out there where they could get some people who work in the field. Although we've had...
there's not really an infectious disease link.
No.
Although when we've had Ian Lipkin on, he's talked about it somewhat, and he does work on it.
Well, he has personal experience, but that's different.
He does work on it, though. I think he's done some studies, and he did talk about it in an intelligent way, but not just briefly, of course.
Well, he is a neurologist, by the way. Don't forget that.
He is, yeah.
Right.
Daron writes, one good gender-neutral word that can be used to replace guys is folks instead of saying you guys. I've trained myself to say you folks when addressing a group of men and women. I work in the 3D games industry, but my hobby is biology, especially cytology and visualization.
Oh, wow.
I listen to TWiM and TWiV regularly and enjoy them immensely. You folks are doing a great work for the world and teaching us about the marvels of the unseen world. All right.
Thanks.
Yeah.
Thank you.
No, thank you.
Folks has come up a number of times.
Thanks.
Sometimes people spell it with a U, though.
Ooh. Alan, you're next.
All right. Just for that, I'm next. No. Nathan writes, Dr. Racaniello and TWiV team, very interesting episode 390. With the increase in arbovirus emergence, we're in desperate need for more efficient methods to catch mosquitoes. The trap that Ethan and Jonathan described obviously sounds really awesome and could help advance the field, but there seems to be a disconnect between the trap and using mosquitoes for non-vector-borne pathogen surveillance. Specifically, detecting viruses left after the boom, with the boom being an outbreak. As briefly mentioned, CO2 traps do not catch blood-fed mosquitoes. Catching mosquitoes that had fed and are resting is more ideal, but for many species, that's a difficult task. A group that I work with at Colorado State University is using Anopheles gambii for similar purpose, and he sends a link. Our approach works very well for a few reasons. Gambii feed indoors on humans, then rest on the inside walls of houses for 1 to 2 days while digesting. We then use giant vacuums to suck them off the walls and then squish the contents of their abdomens onto FTA cards to preserve the DNA or RNA for sequencing. We validated our approach in the lab, and we are currently using this protocol to look for human viruses in Liberia and Burkina Faso. So far, we can say that a lot of people have herpes viruses, and a dog had canine distemper virus. Thought you all would be interested since Ethan and Jonathan did not mention our study. And Nathan is at Colorado State University, Department of Microbiology et al. And Kathy has helpfully posted the information on FTA cards, which are
lovely, fast
technology for analysis. So I guess this is it's kind of high-end blotter paper.
I think so, and it coincidentally came up in a later listener's email, so I was happy that I had figured out what they were. And what he says in his the take-home message of the PLOS pathogens paper is that they leverage the feeding behavior of mosquitoes to sample human blood, which is then preserved on filter cards. And so with that, they can detect vertebrate viral pathogens in realistic field collection timeframes.
Right.
So that's how they discovered their herpes virus and their canine distemper virus.
Why is herpes virus in the blood? I guess Epstein-Barr.
That's what, Epstein-Barr, and what was his other one?
CMV? Or one of the HHVs, probably, yeah.
Oh, rats. Epstein-Barr. He just says Epstein-Barr in the author summary.
Okay.
I read the easy version.
That makes sense. Yeah. No, that's cool. The guys at Microsoft, and they are two guys. It's an interesting technology. It's kind of wowing, but it does have limitations. But this is more labor intensive. You have to vacuum them off the wall, right? It's not automatic. Right. You could make a drone that would vacuum them off, right? Anyway. It's pretty cool. Anthony says, perhaps TWiV can enjoy a day trip too. I wonder if influenza is a concern for the sea mammals at the Coney Island Aquarium. Great TWiP episode. So this is a reference to the TWiP episode where we went to the Bronx Zoo, Dickson.
Yes, I do remember that.
And he sends a copy of the CRC Handbook of Marine Mammal Medicine where he has the page on influenza in whales. So
they do have beluga whales at the Coney Island Zoo.
Yeah, belugas.
Yep.
And so he wonders if we could go and do a TWiV and find out about
why not
whale influenza. Sure. That fellow, Paul Cowley from the Bronx Zoo, he's the head of
yes, he is.
That too. He's head of all of the zoos.
He's the chief veterinarian for all of the zoos.
Tell him to find a viral infection, and we could do a field trip, get everybody back to New York, and we can go to Coney Island.
We could do the Nathans too while we're
there.
there.
Have you ever been to Coney Island, Kathy?
No.
I bet you'd like to.
Oh, you would love it. Kathy, you would absolutely adore it.
It's
great. I would.
It's a fun place.
And the Nathan's hot dogs are to die for.
I was just going to say.
Not from,
can only go
for.
Yeah.
Now, the Cyclone roller coaster is gone, right?
I don't think so.
Did they finally tear that down?
No, I don't think so. I think it's still there.
Oh, I thought I saw something about that it was getting taken down.
They have a minor league baseball team that plays there by the same name.
Okay.
The Coney Island Cyclones.
The next one is from Kevin, who writes, "Hi, Kathy. Ask and ye shall receive."
Aha.
Cheers.
And it was that he sent a link to academic journals by preprint policy. It's a Wikipedia article that just lists them all for you.
Oh, whether or not they'll take a submission if you've done a preprint.
And the various caveats.
So this existed, I guess. And Kevin certainly didn't put it together himself. I hope.
I think it probably already existed.
It says at the top, this article has multiple issues. Please help improve. Wiki is often very difficult. I've lost track of who's next.
I am.
Good.
Todd writes, Dear Vincent, so everybody else drop out. No need to read this email on the show. Okay. As I don't want to take up the team's or your listeners' time. Thank you for reading my note regarding preprint publications. I just wanted to apologize if you felt that my email was targeting your blog post that brought this whole issue up in the first place. My comments were meant more generally. As a blogger myself, I tend to do keyword hyperlinking, as Alan suggested. Although if I am discussing a particular paper, I will try to indicate the author, title, and journal so readers can find the paper in case the link breaks sometime down the line. I haven't encountered the preprint publication dilemma myself yet. Thanks again for all you do and the rest of the TWiV team do. And then he says, P.S. 2 rules for the internet. Number 1, you are wrong. Number 2, if you're right, please see rule number 1.
That's great. I love it. I love it. It's true.
Yeah.
This has a long time ago when we went through the preprint paper on dengue antibodies enhancing Zika infection. No, it wasn't you. It's okay.
The cyclone still exists. And
yes,
in 2015, it was listed as the 16th in the top 50 wood roller coasters.
It's wood.
In Amusement Today.
Is wood. And
Greeks. Yes, there was a different one, a different historic one there that I think that they closed.
Look, we only have 3 emails left today. Wow, there are a lot more.
Zephyr.
Let's finish
Zephyr was.
Let's finish it. There are 2 more on the Microsoft stuff. Alan, you want to take one?
Yes.
Scott.
So I've got Scott. Scott writes, Dear gents.
Oops.
Hey!
Okay.
That's bad.
That's bad.
Guys is bad enough, but
Guys, I don't mind.
Yeah. A friend shared your web link to me, and I have to say I really enjoyed the banter. Anyway, I'm part of a team that is obsessed with building a better mozzie trap and found the discussion of the new drone traps fascinating. For the sake of your virological interest, we've been beating down the bottlenecks on a trap system designed to detect arbovirus activity in Australia for a few years now. Beating down the bottlenecks. I like that. That must be an Australian thing. Basically, we use honey-soaked FTA cards, nucleic acid preservation cards, that mosquitoes feed on and expectorate any viruses. And he links to a manuscript. This eliminated the need to pool massive numbers of mosquitoes. Second, we developed passive traps that act like minnow traps for skeeters. The beasts follow the CO2 plume up through a funnel into a crate from which they can never escape. This eliminated the need for motors, fans, and batteries that people like to steal. The FTA cards are housed in the crate. Third, we've tried to make the trap a mozzie hotel, providing bed and water, so we can keep them alive and get repeat feedings, all the better to detect virus. We termed this system the SMAC, pun intended, Sentinel Mosquito Arbovirus Capture Kit. Okay. The system currently captures mosquitoes and could be a replacement for sentinel animals. Attached a picture of 80,000 mosquitoes we caught near Darwin just for fun.
Good heavens.
That's Darwin, Australia, not the naturalist. One big question, we are often at the limits of detection. The CT scores in the upper 30s using TACMAN. Anything more sensitive in the pipeline? Cheers, hope to hear from you. And Scott is a professor at the Australian Institute of Tropical Health and Medicine in Queensland.
Right.
Yeah, that first link is the photo of 80,000 mosquitoes.
That's a lot of skeeters.
Ooh, it's all over the place.
Yeah.
Wow.
Just for fun, huh?
Yum.
It's interesting. I guess a lot of people are trying to build better mosquito traps.
Mosquito traps.
Absolutely.
Yeah.
And if they do, they'll beat a path to your door.
The next one, Kathy, is from Tom.
I
No good?
totally lost track of where I am. Tom writes, I'm not sure I understand their device, but regarding the Microsoft project to gather blood samples from mosquitoes, etc. with their device, if their device is stationed in my neighborhood in Miami or Mobile, Alabama, they gather a bit of my blood, then they have information about my blood, the viruses in my system, my genetic diseases, etc. Are there privacy problems here? Can people enter the names on the Do Not Gather My Blood Sample registry? What if residents in some neighborhood in Miami do not want their blood samples gathered? Tom?
Well, mosquitoes gather them all the time, of course.
I believe the culturally appropriate response to that in either Miami or Mobile is to shoot it.
The machine.
The machine.
Yeah.
Yeah.
Gather ye mozzies while ye may.
Are there privacy issues here?
Oh, could be. This is kind of territory that hasn't been explored yet. So if they are successful in collecting the blood samples and they then start sequencing that, people could object. On the other hand, my understanding of the law is that if you leave your genetic material in public, it's public. So people have swabbed the New York City subways, for example, to study the microbiome of it. And one of those projects sequenced not only the ribosomal RNA to categorize the bacteria, they also sequenced the human DNA in the samples that they got, and they were able to track various markers through that. Now, that's obviously a very, very pooled sample.
Yeah.
So I'm not sure that there's a legal problem there. There could be a huge public relations problem there, particularly since this is Microsoft, and people have a lot of baggage with that company. So, yes, if they're collecting these samples, I think ethically it would be important for them to make sure that they're looking at pooled samples, so that you wouldn't be able to dissect out that somebody has this or that gene in that particular area. But these are also automatically anonymized samples.
But presumably they could say, they could, if they had enough sequence, say who it is, right? If they had a sequence to compare it to. Yeah, but that's the thing. Your sequence is not out there. They're not going to know who you are, right?
Right.
And I mean, even though one day everyone's genome will be sequenced, that will not be public, right? That will be private medical information, right?
Ostensibly, yes.
And the Russians can hack into that too.
And then who gets access to that? But that's a whole other can of worms that's separate from
everything will get hacked eventually. Here,
Yes.
Did you hear Dropbox, one of the nice finest tech companies?
Yes.
They got hacked and lost 60 million passwords, and they played
Inevitable.
it. And they played it down.
Yeah.
No wonder I couldn't use my Dropbox.
Yeah, you have to change your password. One more email from Bob. He writes, the BBC correspondent Malcolm Brabant attributes his descent into madness to a routine yellow fever vaccination. And he provides a Telegraph article on that, how a jab plunged my life into madness. And Bob writes, I myself, along with my entire family, was given this vaccination as part of a job requirement working in Saudi Arabia. Bob, and I assume that you are okay, because it turns out it is a known complication. with this vaccine. Severe problems include severe allergic reaction, 1 person in 55,000. Severe nervous system reaction, 1 person in 125,000, which I presume is what Malcolm had. And life-threatening severe illness with organ failure, 1 person in 250,000. More than half the people who suffer this side effect die. They put a note, this is a CDC page, these last 2 problems have never been reported after a booster dose. Yeah, because they died after the first dose. No, that's not true.
Well, no, if you survive, the
yes,
point is,
I know.
if you survive the first dose, you're fine to get the booster, most likely. This article is interesting. And yes, it's entirely possible this was a vaccine complication. This particular vaccine has issues, which are known. And that's why it's not part of the standard childhood vaccine series for everybody, because this is a disease you only find in certain parts of the world, and there are risks to getting this particular vaccine.
And it's why we've brought up several times when people are making vaccines on the yellow fever virus backbone that there could be issues.
Yep.
There could be issues. Yep. This is not a perfect vaccine, and it's imperfect in ways that are kind of disturbing. 1 in 125,000, 1 in 250,000, that's not a lot of people, but it would add up if you went vaccinating whole population. And this article raises some very good points. One of the things that came through to me that's not an emphasis of the article, but it really is, I think, the worst part of this is the stigmatization and lack of access to care for mental illness. Mm-hmm. Because what manifested itself in this guy was florid psychosis. He was running around saying he was the Messiah or that he was the devil, alternately, depending on what time of the day it was, apparently. So yes, he really had a serious problem, and apparently had a very hard time getting to care. And there could be a lot of reasons for that. Mm-hmm. When he did finally get proper psychiatric care and they medicated him and brought the psychosis under control, and then he was able to gradually wean himself off the antipsychotics and now has gone back to his job, but that it took several years for him to get the appropriate care. And I think that really is a major problem with this. Yes, the vaccine could very well have caused it, but in addition, we need to do a better job of taking care of people who have these types of conditions, regardless of how it happened.
Mm-hmm. Have we all had the vaccine, by the way?
I did, yeah.
I've had it.
I did it when I went to Brazil, yeah. You're probably for Brazil, right,
not.
Kathy?
Yep.
Mm-hmm. I went to Kenya and I had to get it.
I have not had it.
I am the devil, Dickson.
I do know that.
The very last line of this article says what Alan just said, too, that no one rolls out the red carpet to welcome back people who have suffered a mental illness.
Yeah.
Yeah.
Unfortunately, that frequency, 1 in 250,000, is more frequent than the risk of paralysis after the polio vaccine, which is, considered unacceptable. That's why we're switching away from it.
But the mortality rate from yellow fever is around 30%, so that's a good reason for giving
yes.
the vaccine.
Yeah.
Right.
Okay, let us do some picks of the week. Alan, what do you have?
I am picking something that has a deadline coming up. If you have beautiful graphics from some research that you've done recently, you should enter them in the 15th running of the VISIES Challenge. This is the Visualization Challenge by the National Science Foundation. I think we've picked the winners of this in past years. If we haven't, you can go to the website and you can browse through the winners, which are, in many of them are just absolutely stunning images and some videos. Really cool stuff that people have done that show interesting concepts in cool ways.
Neat. I don't have any good stuff to submit. Do you, Dickson? You said you had a nurse cell photo that would have been nice.
It would have been. I got it. Actually, a nurse cell did win the Small World contest.
Wow.
So that was the
no wonder you're
prize
wealthy.
winner. I didn't take the picture. No, Eric Gravet took the picture. I just supplied the material.
Dixon, what's your pick?
I have a publication from NASA which features all of the science that had nothing to do with the space program per se, that was generated by the space program. So this is called Spinoff. And the magazine features at least 50 of these commercial technologies whose origins were in NASA missions, and they themselves became progenitors of industrial startups, which later on led to big deals in some cases. A lot of it is computer-based and that sort of thing, but it's an interesting way of justifying NASA's budget.
This is great. We should do this for virology.
You know? Mm-hmm. No, that's true, too, because I think that, when you study cell biology because you're a virologist, you might discover something about the basic workings of a cell that wasn't known before, because this virus takes advantage of that unique thing. And I think the interleukin stories have all been generated by people interested in specific infections. But in general, the whole thing relates to the network of how the host handles all of these things. So this is cool. This is really
and this would be nice to know, so that you could cite something other than Tang.
Yeah.
Exactly.
The breakfast drink.
You got it. No, that's true. So when I saw this,
they've got endoscopes, new aircraft engine designs, water mapping for flooding, all kinds of stuff in here.
And it's got the whole archives there too. It goes all the way back to 1996 in this case.
So they have a biomedical application, the process called CELEX, the selection and enrichment process of nucleic acids. It was research supported by NASA.
By the way, I must, as an addendum here, I must say that history was being made I believe it was last week, or the week before that. Somebody on the International Space Station actually did a DNA sequence, the first one ever done in space.
Yeah, they had a little machine up there, huh?
Yeah.
Mm-hmm.
Yeah.
That's cool. There's one now about the size of a harmonica. That's probably what they had. It's called a Minion.
A Minion. That's exactly what they used. That's exactly right. They did use it.
And you just, has a USB port on one side, you plug it in, and it just downloads the sequence.
That's amazing. Just
the sequencing dongle.
Oh, wow. This is quite a magazine.
Yeah.
They do one every year. Wow, it's beautiful.
Glad you like it.
Dickson, by the way, I just took your picture behind the mic, and I put it on Instagram, and I said, here's Dickson recording TWiV, and someone wrote, "Love you all. You're the best. Thank you." Isn't that nice? That's all of us. All of us guys and gals.
Guys and doll.
Kathy, what do you have?
I picked an app that I was introduced to when I was on my trip in Europe. It's called Maps.me. It's a free app, and the good thing about it is that it works in airplane mode. You can download the maps for the area that you need when you're connected to Wi-Fi.
Nice.
And it works based on the quality of the maps that go into it. And I think, for instance, for England, they're the Ordnance Survey maps, which are incredibly detailed. But I first started using it on the bus when we were doing a social outing from the meeting, and I was sitting next to Jane Flint, and she got to watch me starting to use this app and figure out how it works. And I did have to teach myself a lot. It wasn't all quite as intuitive as some other map programs, but you can find out your speed, your elevation. When we were in England, it just had all kinds of detail. It works here in the United States too. And so I highly recommend it if somebody's traveling, download some of these maps and use this app. I had nothing but good things to say about it. And then in the beginning, when you guys were talking about 30,000 feet and so forth, I was looking for the screenshots that I took when I flew from England to Amsterdam on my way home. And when you click on it and it's telling you your speed, if you're going slow, very slow, it shows you a turtle and then a walking person and a bicycle and somewhere in there is a tractor, and then it changes to an airplane, just the nose of an airplane and a full airplane and the altitude. And then somewhere above 28,000 feet, it stopped registering. And then I started taking screenshots again as I came down. And the really cool thing was that when I landed in Amsterdam, the altitude was -10, -12, -15.
Yes.
So that was just really fascinating. So
It's
I
cool.
recommend that you check out this. And if you're in Amsterdam or somewhere in Holland, you'll be able to see that you're below sea level.
That's very funny.
Yeah.
That's cool.
Downloading it right now.
And I emailed you guys the screenshots of my trip, so
Cool.
you can check that out.
Nice.
All right, I have a pick that is a book. It's written by a science journalist for the New York Times, Donald McNeil, and it's called Zika, the Emerging Epidemic. That
was quick.
Yeah.
Indeed.
And it's a short book. You can read it very quickly. Norton sent it to me. And it's he's been writing about Zika a lot. He also wrote about MERS and Ebola and so forth, and had a little exchange with me by email several years ago over MERS, in which he said, I'm a journalist, not a scientist. But anyway, it's a nice little book if you want to sort of go over the history of Zika. Pretty easy to read. And he has some interesting stuff. I was just reading a chapter where he interviews someone at the CDC when they had said they would not make a firm statement about women living in Zika-infected areas. And he interviewed someone from the CDC. This is particularly in Puerto Rico. The doctor said, we need some firm guidance from CDC, and they weren't providing it. And the woman from CDC said, well, we don't want to interfere in people's lives. Oh, my gosh. What do you need? CDC, that's their job.
They're not interfering in people's lives. They're just giving advice, and they do that all the time.
Telling women what to do in the midst of an epidemic is difficult, she said. Anyway, that's a really good exchange in the back of the book. But anyway, it's a cool book. Check it out, Zika, the Emerging Epidemic. We have two listener picks, one from Tarwin, who picks Darwin's Radio by Greg Bear, a great sci-fi romp that stars virus researchers and the CDC. Even though it goes further than what is likely in reality, it does have some very interesting thoughts on evolution and endogenous retroviruses, and the science seems pretty solid in most places. Thanks again for all the work you all put in, regards Tarwin, programmer, etc. This sounds familiar. Has anyone read it?
No.
Mm-mm.
Darwin's Radio. No. Okay, well, we'll check it out.
Is it on the TWiV bookshelf?
Yes, it will be. It will be on the TWiV bookshelf. What is that anyway? You mean
It's in Amazon.
Yeah, I'll put it on this weekend. And Bowden is back with his pick. I would like to contribute a pick of the week in your honor. This was also the start of something big. Scientific journals back in 1665. And he said a link to the Philosophical Transactions of the Royal Society. And that first issue back in 1665. Wow.
Gee whiz.
Wow. It's incredible.
The table of contents. And then you click through, and then everything's in this old English typeface, but it's there.
Observables upon a monstrous head. Oh, cool. That's very neat. This website also provides the very first advertisement in the very first issue of a scientific journal. And he provides a link. Advertisements have come a long way since 1665. I'm waiting for it to load.
Was that a plague year?
And that's what you put down here, Kathy, right?
Yeah, it is.
You can read it.
So, well, you can finish his letter. You two.
Oh, you two have started something important, free, and nearly instantaneous. That's cheers, Bowden. Thank you.
So what he posted is really more of a notice than an ad, and it says, the reader is hereby advertised that by reason of the present contagion in London, which may unhappily cause an interruption as well of correspondencies as of public meetings, the printing of these philosophical transactions may possibly for a while be intermitted, though endeavors shall be used to continue them if it may be. And what it's referring to is the Great Plague.
Right. Right.
May be interrupted.
Yes.
And that's all in the 17th century text. You know, it's an advertisement.
Right.
Yeah, it's here. Finally, it loaded here. You could click on that and see.
By reason of the present contagion. Yeah.
Thank you, Bowden. That's very nice.
Those are all S's, Alan.
I know. But it's so much more fun to pronounce them as F's.
Were any famous English scientists listed in the contents for the first issue?
They didn't list the authors on the contents page. I don't think. Did they? No, they just. There
weren't any scientists in 1665.
Well,
there was
Robert Boyle and Henry Oldenburg.
Boyle, not bad.
How about Newton? When was he around?
He was there.
David Thomas,
later.
Walter Pope, Dudley Palmer, Edward Digges. So I take it back. There are some names, but not on all of them.
Right.
Mr. Hook.
Mr. Hook.
Is that Robert Hooke?
The cork guy.
No, it doesn't.
Did a lot of drinking, so he looked at cork a lot.
And this will be. This is when I'm going to tell you about our book giveaway contest. So if you listen till now, Princeton University Press has just published Virus, an Illustrated Guide to 101 Incredible Microbes by Marilyn Rusink. We decided last time we should have stopped at an illustrated guide. They've given us 3 copies we're going to give away to listeners for the first contest, and this is the second time I'm announcing this one, so that'll be the last time, and then we'll do something different. Write in 200 words or less which virus you would like to be and why. Send your entries to TWiV@microbe.tv. We've got about 4 or 5 entries. They're good. We'll have 2 other ones, which we'll think of in the next weeks, but if you want a free copy of that book, just write that up. And that will be TWiV 405. You can find it at iTunes and microbe.tv/TWiV. And I know a lot of you don't listen at iTunes any longer, but if you would go there, if you haven't ever done it, and just rate the show, you have to find our listing, which you can do through the link on the webpage. Just give us a rating, 5 stars. And that helps new people discover us, because a lot of people still go to iTunes to discover new podcasts. And if we get a lot of ratings, we're up there on the listings, and people will see us and learn all about how cool viruses are. Please send us your questions and comments to TWiV@microbe.tv, and consider supporting us. Not just TWiV, but all the science shows at microbe.tv/contribute. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
We have a new PhD student here who just joined us from University of Michigan.
Oh, good.
So she should be great.
Absolutely.
Dickson Despommier is at verticalfarm.com, and soon to be another website, which we'll announce at some point, right?
Probably next TWiV.
Pretty exciting.
Yeah, I think so.
Thank you, Dickson.
You're welcome, Vincent.
Alan Dove is at alandove.com. You can also find him on the Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsors of this show, CuriosityStream and Drobo. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.